Today, I want to show you some tools for working with protein sequence alignments. So we'll talk a little bit about how you can get these alignments with tools like Clustal Omega, which you can access through UniProt directly, sometimes after a blast if you're trying to find related sequences, and then how you can display them either interactively with a tool called JalView, which has a web server as well as a desktop version that you can use. And if you want static pictures, such as for a publication, maybe, what you can do is you can actually use this tool called ESPSpript, and it will generate nice looking figures for you. These figures you can also then adapt, so you can modify them, you can annotate them further in a tool like Adobe Illustrator or Inkscape, which is a free alternative to Adobe Illustrator. And they also have a bunch of ways that you can do it directly in the program as well. And finally, I'll introduce you to a tool called ENDscript. And so ENDscript is really cool. It basically is by the same people as do the EPSpript. And what it does though is it'll also put like structural information from PDB files. So it'll make an alignment and then it will generate PyMol files. So you can actually get graphical representations. And so it shows the conservation of the various residues, as well as you have one where it is kind of like this sausage style, where the thickness of the sausage indicates kind of how different the structures are in those regions. So the regions that are bigger sausages, that's regions that are more flexible, or they're just in different locations in different structures, as opposed to regions that are kind of skinny. And so the skinny regions too, you'll see a lot of them are going to be well conserved with that red. And these you can then do things like overlay with the substrates that you know, and you can see that those actually align to the active site. So that's pretty cool. You can see which ones are identical. You can do all sorts of various things, see the sites from the PDB file because it integrates all that PDB information in. So really, really cool stuff about that. But that's kind of like icing on top of the cake. So let's talk about how you can just deal with simple sequences to align. This is not meant to be a tutorial in any way. This is just kind of showing you what these tools are, when to go to what, and why. So this is Clustal Omega. So you can access Clustal Omega. It is through the EBI. You can access it directly. You can also, if you have, it works for DNA, RNA, and proteins, as well as like JalView, you can be visualizing DNA, RNA, proteins sequences with JalView. All these tools, by the way, are totally free. And so the Clustal Omega is good for multiple sequences, like more than two. If you have only two, then you can use the pairwise sequence alignment tools. Often I'm accessing Clustal Omega or pairwise sequence alignments directly through UniProt. So UniProt's like your go-to place if you want to know about a protein or you want to find information about a protein, links about a protein, all this various stuff. So say I'm interested in human MDH, so malate dehydrogenase. And for some reason, the first things it gives me are lactate dehydrogenase. But here we go, malate dehydrogenase. So you can see there's a cytoplasmic version and a mitochondrial version. What I want to do is I want to actually compare these two. And so I can click on them both. And what I can do is I can add them to my cart and then access my cart over here. Or I could just go directly and I could go to tools and it's going to give me some options. Right now I want to do align, and later I'll show you what I'm going to want to blast. So blast is going to compare your sequences to other sequences to find related sequences, whereas align is just going to take the sequences that you give it and align them. And so when you have jobs running, you'll get this kind of like screen that looks kind of like this, and it's going to show you all of the jobs that you have running or that have been done. You can move away from this page. And your results, you can always find your results over here in this little like closed pursey type thing next to the basket. And don't worry, they're not going to charge you for anything. The basket's just kind of sequences that you want to save. When your results are ready, they will show up here. And so for the sake of time, I just have the results here. So it doesn't take that long, but my computer is a little overwhelmed right now because I have a lot going on it. But you'll see an alignment like this. And you can do things like see trees. So it's not that interesting because there's only the two sequences right now, but you can see percent identity matrix. And this is going to tell you about sequence identity. So whether the residues are exactly identical and how many residues are exactly identical. Then eventually you should see some sort of tree that's showing you how identical the sequences are. And if you had more than two, it would look a little more interesting. For now, let's just go back to the overview. And you can see that I could highlight it by different properties. I could select various annotations. I could do cool things, but it's not that interactive. And it's not really giving me an option if I want just like a nice figure. And so typically if I have something like this, I just do a screenshot. But I can make a very nice figure of it using EPSpript. So what I want to do is if I go to download, what I can do is I could download the resulting alignment in Clustal format. So not the one with the numbers, but just the Clustal format. And then it's going to take me to a screen with the results. Now, what I want to do is I want to actually just save this. So if you just go to save as, it'll save it as a text file. And then you could actually open that text file directly in your EPSpript. So if I come over here to my EPSpript, now what I can do is I can actually just choose the file and I'll choose that Clustal file. And now EPSpript, what it's going to do is it's also going to overlay the structural information. So it's going to give me something like this, where it's overlaying the structural information. And note that you can make like really, really pretty versions of things too. So if you go to gallery, you can see there's a bunch more complicated things you can do, where you can do things like color things different colors and indicate various important residues. And so sometimes though some of that stuff is just easier to do after the fact in a vector graphics software like Adobe Illustrator or Inkscape. You can see there's like different levels of beginner, advanced, expert, and they have really good documentation about how to actually use this software. So I'm not going to go into all that details. The minimal thing you need to do though is you're going to need to choose a PDB file. And so you can search the PDB. For this, you're going to need to actually know the PDB entry code, or you can have your own PDB file. So say you have a AlphaFold predicted model or something, you can use that. And then you would just upload the file as opposed to doing this where you're going to find the file. And so here what I did was I uploaded that alignment I had. So that was my Clustal file. And now I search for the PDB file for WLO, which I know corresponds to mitochondrial malodehydrogenase. It's going to give me some different options depending on how I want to color things. Note that here I told it to display I want to do the mitochondrial malodehydrogenase before the cytoplasmic because the structure that I uploaded goes to the mitochondria. So that's why I did this 2-1. So here it tells you a little bit about how you can do this stuff. Anyway, now you get to the fun stuff. You can kind of tell it how you want to color things and format things, the font size, all that good stuff. When I press submit, a pop-up window is going to appear. Note that you need to have disabled pop-up. Need to have enabled pop-ups, so don't block the pop-ups. And once you unblock the pop-ups, you need to refresh the website or else the results won't show up. I could exit out of this window and then I could get it again if I come over here to results. If I go to doc, it's going to give me the documentation to tell me how all of this works. Over here, you can see I have a PDF file and a PostScript file. PostScript is kind of like PDF. And so either way, what I'm going to get is I'm going to get a screen, or I guess if the PostScript, it just goes directly to my downloads. If I wanted to, I could open this up in Adobe Illustrator. And now I can easily edit it, and I could add annotations, anything like that I want. There are, again, also options where you could do things. If you go to the expert mode or even the advanced mode, there's a lot more options that you can do with this and tell it how you want to color things. You could color things by the physiochemical properties. You can replace sequence names. You can do all this stuff, and you could get the PDF files. You can get the PNG files, TIFF files, that sort of thing. I typically get the PDFs, and then I could further edit them. Or I could, the PDFs, if you blow them up, they're still going to be nice and big. But for some things, you might want the PNG or you might want the TIFF. And TIFFs are higher quality than PAGs typically. So you can see these are higher resolution, but they're going to be bigger files. So just depends on what you want. But you can get something pretty like this, where you're indicating the secondary structures. You've got your alpha helices, you've got your beta sheets, you've got your turns, and that sort of thing. And again, they have a whole like user guide about what means what. Okay, so that is that. That's not that exciting of an alignment. And so you could do much more exciting alignments. And so what if I were to go back to UniProt? And again, I want to go to that human MDH. And it's actually in my cart, so I can just go to my cart and get it directly there too. Or I could have saved it at this point, whatever. Anyway, I want to go to it. Now, the entries, I have more on UniProt elsewhere. There's a lot of cool stuff you can find in a UniProt. And one of the cool things that you can find in a UniProt is the ability to go to this tools and blast. Now what I do is I select blast. Blast is going to search it against the database of proteins, other proteins. So you can find proteins that are similar. You have the option to kind of like restrict it to various taxons if you want. You can do things like choose the threshold, the databases that it's searching. So by default, it's doing this UniProt KB reference proteomes plus prot. I could have it do all sorts of different things. I'm just going to change the number of hits down to 50 so it doesn't take forever. And now run blast. I'll get something like this now. And so you can see I have a number of different sequences. And it's showing me things like their similarity. If I want, I can kind of change this. So maybe I don't want identical sequences. I can go to 95 or something, and now I'm getting ones that are similar, but they're not identical. What I can do too is if I click this, it's going to select all these, or I could select just a portion of them. And then if I go to tools, I can do this align the selected results. This is going to give me an option to align things. And then I could go and I can align them. Before I did an alignment with 50, and so these are the 50, and you can see that this is, okay, that's kind of cool, but it's going to be really hard for me to navigate through all of this. So instead of going and looking at it like this, what I want to do is I'm actually going to open it in JalView. So you can look at JalView, you can use JalView from either the web version or the desktop version. So here's the JalView. And you can see that right now it looks pretty boring. But there's actually a lot of really cool information. It's just not showing. So if I go to color, right now it's not coloring by anything. If I go to Clustal X, well, now it's going to color kind of by its properties. And if I go to color and I do one of these scores, well, now it's going to color it based on kind of like how similar the residues are to one another. And so these structures are all pretty similar. And so that's why you can see a lot of it's identical and a lot of it is going to be really similar. And in fact, if you come down here, you can see conservation. And so when the bars are at the top, that's basically they're all identical. And you can see they also have this little asterisk under them. But when it's lower, well, now these are less conserved. You can also kind of do things like if you want to color it by hydrophobicity, you can color it by strand propensity, by all sorts of different things. One cool thing too is that say I want to go and I want to color it by percentage identity. And so that's the sequences are exactly identical there. And now what I can do is I can come over here and I know that this MDHM, this is the malodehydrogenase of human. What I can do is I can look up the structural data for it. And it's actually going to map that on. So if it doesn't have a structure, it won't have anything to map it onto. But if it does, it's here. And so you can see it's actually mapping that data onto here. And so you can see that the ones only like all these that are dark blue, those are identical in all these structures. And the ones that are lighter, those are some of them are different. And again, this isn't a very exciting alignment because these are all so similar. But you can do things where you have a lot more sequences or you have varied sequences, and then you could get some real variation. So here I'm showing you the same structures but this time in the desktop version of JalView. Here they're colored by Clustal. So again, by their properties, but you could also do it by various other things like conservation, things, whatever you want. You can alter things. And you can do things like change the name of the sequence so you can edit the name of the description. And there's a lot of different options that you can do, lots of different things. I'm not going to try to do a tutorial on this right now or anything, but they have good resources on their website. Speaking of resources on their website, if you go to EPSpript, so they actually have this help online and web guide is what they have like all the detailed information, and it tells you like, okay, well, this is what this means and this is what this means. And so these are the matrices you can use to determine whether things are how similar things are. And then you have like a red box is saying they're identical. You have red characters, they're similar, and then they're similar within groups, so they're similar across groups. So if you have any questions about any of that, the place to go is going to be that user guide. There's also these tutorial and examples. So they have tutorials taking you through both beginner and advanced things. So you can make really pretty figures. But you can make even prettier figures using this tool called ENDscript. Now, what ENDscript does is it's actually going to do like that blast search based on a PDB file, so based on a structural file that you provide it, and then it's going to map that information onto the structure. And it's also going to pull in structural information from PDB files from crystal structures, cryo-EM, all that stuff. The PDB files that are all those PDB files is going to find PDB files for the structures, for the sequences that it finds are similar to your protein. So it's going to look and find similar sequences. It's going to find the PDB files for those sequences, and then it's going to map all of that information both into one of those EPSpript kind of looking figures, one of these things. There's 2D figures, as well as it's also going to provide these 3D figures. And so you'll see that you're going to kind of get that original figure that you just got from EPSpript, but here you're getting it from this other program, which is related to EPSpript. They're like connected. Here you have just the one you inputted. And here now you have the alignment. So it actually does an alignment now. It goes and it searches, it does one of those blasts to find sequences that are related, and then it finds the structures of those related sequences and actually overlies the information about those structures on top of the sequence alignment. Down here, you're seeing solvent accessibility and hydrophobicity. Here you're seeing the sequences. You're seeing when they're identical, the red, when they're similar, and when they're not. And you can actually go and you can change the how you want things displayed. It'll take you back to EPSpript where you can do all of those modifications that you could do before. But now it's also going to map that information onto the structure. And so you can see where there are conserved regions of proteins, that's going to be red. And when they're gray, that's not. And so here are just some of the figures that they had. It has a similar looking interface to what you saw with EPSpript. And again, they're by the same people, so it makes sense. Here you're going to choose a PDB file just like you had before. So you could either choose a PDB file that's actually published in like the protein database, or what you could do is you could actually go and upload your custom file. So again, like if you had an AlphaFold model or something like that, you could enter that here. You have a structure that you just determined with X-ray crystallography, so it's not published yet. You can choose that here too. You can also make different selections, again, kind of like how you could have things that were kind of simpler or more advanced. And see, you can see here it's going to do a Clustal alignment. You can choose a database, choose how significant the results you want to be. Right here, it's discarding identical sequences, which is nice. I can have it do things like I can change the font and columns. Again, that's going to be for the alignments, but it'll give you those options to make changes to the alignments later. When you export it to EPSpript, you can export it to EPSpript and then make all those changes there if you want. When you run this, similarly, you're going to get another one of those pop-up windows. It's going to give you a bunch of files. Now, some of those files are going to be the alignment pictures, so you have the static ones. You also have the links to the PDB files that it found for the structures, and you can download those PDB files if you want. You can also get a link that'll take you directly to JalView for that alignment. And what you have is these PyMol files, and there's two PyMol files. One of them is going to be, so PyMol is a structural visualization software. And so one of them is going to be just kind of like a normal looking cartoon, except it's colored by its conservation. And another one is going to be let me just turn off the stuff that is not, the stuff is just like a sausage. And so here's a sausage where it's showing if it's a thicker sausage, that means that those regions are kind of, the positions of those residues are more variable in the different structures. And so that might indicate structural flexibility, or that there's kind of like just variations in the structure. And so the regions that are really well conserved are often going to be less variable, although you might have regions that are well conserved at the residue level that kind of have more flexibility in them for the sake of the protein functioning and being able to kind of like be dynamic. But if you have structures that are very similar in their sequence, they're often going to have very similar structures. And so then they're going to have the structures are in the same place all the time. And so in all these structures, in all these different isoforms, and so that, or in homologs, then so they're going to be those thin lines. So that's the sort of thing that you can tell here. And because it gives you that pymal session and not just figures, what you can actually do is you can go and you can do things like map in the position of substrates that you know are there. You can also, even without that, it gives you the options to, depending on the PDB file you input and how much information the PDB file had, you could do things like look at the bio unit. And so this was like crystallized with four subunits, but you can see that it's really only a dimer. These ones aren't connected. And so, but you can see that this is showing this one subunit, and you can see the broader context. You can also do things like look at identical residues. So in all the structures that were aligned, these ones were identical. It pulls in the sites, and so you can get the sites if things were like marked as sites in the PDB file. So like this is the active site, you can see the active site here. You can also see the solvent surface, which is again colored by accessibility. And so really cool stuff. And it gives you the pymal session file as well as the pymal script, so you can go and do it yourself. So again, all these tools are totally free, and they have really good resources and documentation. So if you want to just do a simple alignment, you can use Clustal Omega. You can get to Clustal Omega or similar tools directly from UniProt. And so you search UniProt for the protein you want. You can align the sequences of those proteins. Then you can, if you want, you can then open that in JalView. So download that Clustal file. That will be on your screen. And so depending on how many sequences you have, it'll be a lot or it'll be a little. But you can go ahead and download this, and then you can open it in JalView. And you could do that in the web server, or you could do it in the desktop version. And so it's easier to use with the desktop version. It's free too, but it's easier to use. Like you can do more things with it, and you can kind of navigate it more easily. But you can just quickly go to the web version. So JalView JS is the web version. Again, these are both totally free. If you want a static picture for some publication or maybe for a report, something, EPSpript is your place to go for that. And it's going to give you a lot of different options. And they have a really good documentation about kind of how to do things and what things might mean, as well as these web tutorials, which are really helpful. If you're wanting to do things with like structural information and mapping the conservation onto the structures, pulling in information from PDB files, well, here you want to turn to ENDscript. ENDscript is really, really great for this. It'll give you those pymal files, those pretty pictures you can play around with, as well as just kind of like helping you find PDB files of related structures. So lots of really cool stuff, and hope this was helpful. And again, this wasn't meant to be like a tutorial. They have great tutorials online. This is just more like, okay, where do I start? Where do I go? And just know that these things are out there, because there's a lot of cool stuff out there that sometimes people just don't know about. So now you know.